Sorry, that cut out for some reason. So we were able to replicate DNA using the plasmid. DNA fragments are usually pretty small. They come from bacteria. They're extremely small. And so one way we can clone bigger DNA fragments is by using something called the yeast artificial chromosome vector. You'll just be able to get a really big chunk of human DNA. It'll go through the same process. It will be cut by the restrictive enzyme of eCore1 or whichever enzyme you want. You'll cut it, and then the human DNA will be able to be put onto the plasmid. Yeah. Okay. This section will be about how we can clone all of the DNA that's formed from an organism. And so you have this human double DNA strand, and you'll be able to cleave it with something called the restrictive nuclease. It'll be able to produce millions of DNA fragments, and then those DNA fragments will once again be put onto the plasmid. We're using the restrictive enzyme. You can see various of the plasmids right there in little circles. And then we need to make sure that all of these DNA, all of these plasmids have been cut with the same restrictive enzyme once again, because they all need to, when they get combined to form the new DNA strand, they need to find a complementary sequence. They can't be formed by just putting two DNA or one, something like that. Nucleotide, my bad. And so once you have all of these recombination of the DNA in the plasmid, you will have different DNA fragments, right? Because it's a really big DNA strand. So you have different DNA fragments. And so one thing you could do is introduce plasmids into the bacteria, and then you're gonna have something called the human genomic DNA library, which is basically where it has all the DNA and the genetic information of a human. It's just been sorted into something called the library. And so when you look here, you have 5 different books. One book consists of a plasmid with one specific DNA strand. Another book has a different DNA strand, and etc. Here in this example, we created 5 plasmids with 5 different DNA fragments. So that was able to create 5 different books in the human genomic library. Because the human genomic library, once again, is the full DNA genetic material of a human. It's everything that the human has. Yeah. And then when you want to know about the proteins that are made by these specific books and these sequences, one thing you need to make sure of is that the DNA inserted back into the bacteria does not have any introns. Because once again, if it has the introns, it won't be able to complement itself with other DNA and get its right nucleotides. The introns will affect the forming and the DNA synthesis and all of that. And so the first thing that you need to do is get an mRNA that has its introns removed. And so one thing you can do in this example, you're getting tissues from the brain. You're going to first isolate these tissues, the tissue cells from the cytoplasm by by purifying the DNA, and then you're going to make a DNA copy of the RNA. And so first you're going to start off with the mRNA that has the introns removed, and then you're going to add something called a primer, which is a long strand of thymines. What that will do is make a complementary DNA with reverse transcription. It will proceed with that. Reverse transcriptase is basically an enzyme that's added to the mRNA, which will reverse transcription. It will cause that mRNA to go to DNA, and it'll create a DNA strand. But the thing is, it won't create the original DNA strand. It'll create something called the cDNA. cDNA basically stands for complementary DNA. And so once it creates the cDNA, oh wait, my bad. Yeah. Once it creates the cDNA, it will degrade the RNA with something called the RNAH. And then once you degrade that, you will have a synthesis of the second cDNA strand using something called DNA polymerase. So once you have a degradation of the mRNA, you will be able to create a second strand of the cDNA using the DNA polymerase. And then from there, you can use this back, you can insert this DNA strand back into the bacteria, and it can make a protein by doing the plasmid, inserting this specific DNA fragment into the plasmid using the restrictive enzyme, and then it'll be able to go through the same process over again. Yeah, and this is how you make sure that there is no introns. All right. When we look at, we're going to be now looking at DNA sequences. We're able to use a technology called PCR, which is polymerase chain reactions, in which you can get large amounts of DNA sequences, get many amounts of DNA sequences. And so, yeah, many strands of the one you will work with. So you can use this strand to study a specific DNA fragment. So here you have a double-strand DNA. You're going to use heat to separate the strands, and then you're going to put a primer on using hybridization. And then the DNA synthesis from primers is going to elongate the mRNA, and you'll get various DNA strands. When we look at how the primer adds the nucleotide to the DNA strand, there is something called the deoxyribonucleoside triphosphate. It is basically allowing the strand to extend at the 3' end, because that's the end in which the nucleotides could be added to. So the deoxyribonucleoside will be able to extend that 3' end, and it has an OH, so hydroxide. But when you look at the deoxyribonucleoside triphosphate, that is basically a triphosphate that prevents the strand from extending any longer. So when you look at the PCR, you're adding nucleotides onto the 3' end of the DNA strand. The minute it adds a nucleotide that is a dideoxyribonucleoside triphosphate, it will stop the synthesis from happening. And we're able to find the length of that synthesis, of how long it was able to synthesize for, by using the gel. And I need to explain that. When you look at this well, you have all these different strands. You have all these different pink strands. So when you look at this well, you're able to see that it has the D-deoxyribonucleic triphosphate of adenine. I think that's what it's called. Adenine, thymine, cytosine, and guanine. You look at this well. You're able to tell which one was synthesized longer by the one that is closest to the well. So the longest strands means it was synthesized longer. The dideoxyribonucleotide was added a bit further down. And you're able to tell when it's closer to the well of the PCR. When it's farthest away from the PCR, that means the deoxyribonucleotide was added pretty quickly, maybe like 10 seconds, I'm not sure. It was added pretty quickly. And each specific well indicates which dideoxyribonucleotide triphosphate was added. So in this well, the adenine, the dideoxy of A was added in this well, the T, the C, and the G, respectively. And yeah, one more thing. When you're looking at the plasmid, you're going to be cutting the restrictive enzyme and all of that. But you need to kind of tell yourself, how can you distinguish which one actually inherited that DNA that you want to clone? One way you can find that out is finding out which one is resistant to an antibody. On the plasmid, there is something called an antibiotic resistance gene. It basically means, say if you add ampicillin to a cell, it'll be able to disintegrate the ampicillin. It'll be able to resist the stuff of the ampicillin and its harms. So when you cut the plasmid, you'll be able to, you're going to cut it with the restricted enzyme. You're going to put the DNA fragment, and then when you culture it, you'll be able to put it in ampicillin. When you put the many copies of the plasmid copied DNA fragments, when you put them into ampicillin, you'll be able to shake it. Yeah, you'll be able to shake it in the ampicillin, and whichever plasmid is left will tell you, oh, these plasmids inherited the DNA that is resistant to the ampicillin gene, to the ampicillin, I mean. Well, let me explain that one more time, just for me. So you'll be able to find out which plasmid inherited the DNA fragment that you want to study by stirring all the copied plasmids, into ampicillin. Once that is done, you'll be able to see which ones were left. The ones that are left will tell you that it has the antibiotic resistance gene. And now, to double check even further, you're going to find the right ones that have the specific DNA sequence using hybridization, in which it will the label, the probe of the hybridization will find the right sequence of the DNA that you want to study. It's a very long process. Let me explain this one more time before I end this for me and just to show that I get this. So you have a plasmid that has an antibiotic resistance gene. You're not going to cut it. You're going to cut it with the restricted nuclease. You're going to put the DNA fragment that you want to study, and then you're going to put it back into the bacterial cell. You're going to culture it with the bacteria, and it's going to form various purified plasmids with the DNA fragment that you want to study. You want to make sure that it is studied, that you want to make sure that it was inherited by getting ampicillin, and you're going to get all the copies of the plasmid, stir it in the ampicillin, and then from there, see which ones were resistant to the ampicillin. The ones that were resistant to the ampicillin is the ones with the DNA fragments that you want to study. To double check even further, you're going to be using something called the hybridization, in which you will see which plasmid has a specific DNA fragment by matching the nucleic acid to the hybridization that you set it to. And then from there, you'll be able to find the plasmid with the right DNA fragment because it has the right nucleic acid sequence. Yeah. That's very hectic, but this is very fun. Very interesting. I'd like to end the presentation. Thank you for watching. Have a great day.